So we're moving into genetics lab part 3. There's a lot of alleles that produce a lot of protein variations. And some of the proteins, they're functional, but they're functional to different degrees, so they do slightly different things. So having one protein totally unfunctional being covered by another protein that's functional, it doesn't happen that often. Dominant recessive in human features, not really that common. Most of the time, like with AB blood, you get an allele for A, you get an allele for B, they're both there, is that you get AB blood. In like human height, is that there's not one gene for human height. Somebody did a calculation last year and figured out that if you look at anything that works into growth, that there's something like 240 different genes that you could say apply to our growth. But usually there's like 8 or 9 genes that are major players. Those proteins are major players in human growth. And of course the environment, how much food you eat and such factors into it as well. So there's a lot of complications beyond dominant recessive, very simple. And we're gonna go to another one that's still a little simple, but it's more of a blending of effects rather than dominant recessive. I'm gonna give you an example, and if the example makes sense to you, then you won't have any trouble with the question that you've got in part 4 for the very first question. I'm getting fancy, I shouldn't. So you have flowers of the same species, but they come in different colors. You get red, you get blue. And you decide you're gonna cross the red and the blue. Well, if you were to do that and all the flowers you got were red, you'd know for some reason red is dominating blue. And if you got all blue, you'd know blue is dominating red. But what if instead of red and blue, giving you red or blue, you get purple? Now, if you were mixing paints, one of the ways that you can get purple is to mix red paint and blue paint. And that's exactly what you're seeing here. You're seeing a red allele and a blue allele being expressed together, and like mixing paints, you wind up with a mixed color. But genetically, this is what's happening. 2 red alleles, neither one of them dominant or recessive. 2 blue alleles, neither one of them dominant recessive. And actually, this is what I should do. Now, the most important thing to notice here is you have a trait, you don't have alleles for that trait. There are no purple alleles here. You only get purple when red and blue are there together. And then the question becomes, well, what happens if we cross the purple flowers with each other? Well, here's the deal. You can get 2 reds back together, so you can get red flowers from that mix. You can get blue flowers from that mix. You can get purple flowers from that mix. It actually turns out there's 2 different ways to get purple flowers. So there's actually 4 combinations. 2 reds together, 2 purples together, the first red with the second blue, or the first blue with the second red. And that means there's 2 possibilities for red and blue together. So if you did this cross, about half the times you'd get purple back, about a quarter of the time you get red, about a quarter of the times you get blue. Now, that's the setup for the question, which I will tell you right now, I made up gerbils don't work this way, but what the heck. So you go to the pet store and you get 2 gerbils. And the guy at the pet store swears they're both males, they're both females, but you get them home and before very long, it's pretty obvious from their behavior, you got a male and a female together. They're both gray, but they make babies. And the babies are not all gray. Some of them are gray, some of them are white, and some of them are black. And you need to show me genetically what's going on with the gerbils. So maybe the first thing you want to decide is what allele is not there before you start figuring out what alleles are there. But as I said, if this makes sense, you can always go back over this, you know, it's a video, you can watch as many times as you need. If you go back over this, then you ought to be able to take and fit this to the gerbils. Now the second question in part 4, I talked about already with height. Height is not the product of a single protein, it's the product of multiple proteins, all contributing to a trait that looks simple, right? We can measure your height, pretty simple trait. There's a lot of human traits like that, where the trait itself, when you break it down to what proteins are making it happen, what genes are producing those proteins, there's a whole bunch of them at play. And let me show you some examples of multiple genes working. Back in the 19-teens, 1920s, something like that, a British doctor named Punnett, who was interested in how this genetic stuff all worked, decided that he could represent genetic crosses with what looked like basically a multiplication table. And you don't really need it for the simple stuff, but here's the simple stuff. In a situation like this, you take the A's across, you take the B's down, you take the O's down, you take the O's across, and it shows you all the different combinations you get dividing the alleles up the way that they divide. But the advantage of the Punnett squares was, what if there's more than one gene involved in the trait that you're interested in? What if you look at both types of blood, the ABO and the RH, here, the A could go on with the positive, but also with the negative. The B could go on with the positive, but also with the negative. The O could go on with the positive, but also with the negative. And what happens there is instead of 4 boxes, you wind up with 4 and 4 and 4 and 4. You wind up with 16 boxes. Is every time you add a gene, you exponentially increase the size of the mixes you can get. And this is getting to the answer for the second question in part 4. When you see a simple trait, but it doesn't inherit in anything close to a simple way, you're looking at a trait produced by multiple genes. They call it a multiple gene trait. There's only 2 genes at play here, but there's 16 different ways to mix them together. Add another one and you're up to 64 and then 120, it gets crazy. Maybe it's 32 then. But it's, the more genes you add, the more possibilities, the more mixes you can get. Which is why in a family, people are all sorts of different heights. Even though, you may have 2 tall parents or 2 short parents, doesn't mean they're not gonna have, a kid that actually isn't within their range. 2 short parents can have a tall kid, 2 tall parents can have a short kid, because they're remixing a whole bunch of alleles together. And if you look at all the different boxes they could mix, there's gonna be lots of tall ones probably, but also some short ones, if you got tall parents. But that's what you're looking at, and this is why genetics is way more complex than people tend to want it to be. When somebody goes, oh, well, schizophrenia is genetic, and they go, well, then why can't we figure out you know, how it's gonna run through our family, because we have these schizophrenics. Well, schizophrenic is a multiple gene trait, and we don't really understand what the genes and the alleles are doing, so we don't really know how to trace the mixes. And even if we could, all we could do is give you the odds. That's all it winds up being. So that's it for part 4.